Hi everyone, I'm Tamsin. I'm another student in Kathy's lab, and today I'm going to talk a little bit about drug repurposing, sort of in context of EEF1A2, because that's really where my PhD project comes in. I've been here about a year and a half so far. So I think when a lot of us think about drug development, we think about it in quite a linear sense. So we're probably going to start off by thinking, I've got my disease, I'm going to find out what causes my disease, and then I'm going to find a new drug that specifically targets that. However, we all know that the body is really, really complicated, lots of different pathways, and we're all aware that most drugs have lots of side effects. And that's because a drug isn't going to do one thing in the body, it's going to be interacting in lots of different ways. And sometimes we can exploit those side effect interactions to kind of be the primary reason we can use a drug for another condition. That's sort of called drug repurposing. So sometimes this is very straightforward. Got lots of examples here of all of the drugs I'm showing at the moment are either in late stage clinical trials or are currently being used as therapies for different rare diseases. So sometimes we're going right from, right there, I've got a drug that was originally used to treat cancer, and then it's identified as a possible treatment for Hutchinson-Gilford progeria syndrome, and that's being used at the moment. And I'll talk a little bit later about how we find these drugs, but sometimes this process is a little bit more complex. So it just appeared there. I've got a drug that was originally repurposed to be used in cystic fibrosis, and then is actually later on repurposed again as a pneumonia treatment. And then sometimes you've got to put a lot more information to get these repurposed drugs. So the drug I've just shown there in the middle was originally a possible therapy for Angelman syndrome. That was unfortunately not a successful endeavor, but was later on found that this drug can be effective at treating Fragile X syndrome when you combine it with a therapy that was repurposed from being an insomnia drug. And then finally, sometimes we might repurpose drugs that aren't actually in the clinic. So the drug I've just shown there, everolimus, was not actually ever prescribed for anything, but will have been held in probably like a pharmaceutical company's database, and then is found later on to be useful for tuberous sclerosis and some forms of brain cancer. So I'm going to talk about why we would drug repurpose. So we've got lots of examples where it's worked, but why would we choose to repurpose drugs rather than make new ones? And the big thing is the time and the money that it takes. So what I've shown there on the screen is what a typical drug discovery network pathway looks like. It take a very, very long time. Going to start off with over 20,000 compounds, and at the end of it, we might get one drug. But most compounds that researchers and drug companies look at to start off with will never, ever end up in the clinic. So drug repurposing can cut out a lot of that time. So we're coming in right there about halfway through at that screening and validation stage. So you've cut out a lot of that early research, removed a lot of that time. And that's right where my project comes in, is at that screening and validation stage. That's what I'm looking at the moment. And it can really cut down the amount of time it takes for us to find a drug. So basic drug discoveries can take about 10 up to 20 years. With drug repurposing, can still take a long time because there will be challenges, but you can cut that down for about 3 to 12. And if we're taking less time to discover drugs, we're pretty much always going to be using less money, which is a really promising concept for rare diseases. There's lots of different ways that we can find drugs to repurpose. So where my project is really focused is this known molecular target that I've got there on the right-hand side, because I'm specifically always going to be looking at EEF1A2. And I'm doing that using cell screening models. And I've actually got a picture there of some cells that I've made that have the EEF1A2 protein in them, with lots of a few other proteins that give them different colors that make them quite good for sort of high throughput screening of lots of different drugs. But you can use other approaches, and other people in the lab do, and other rare disease researchers. So you can do this known target screening virtually, and that would be where you might have a computer that's going to predict which drugs are going to match onto certain parts of a protein. And you can use mouse models like Grant talked about. You might not be looking at a particular molecular target, but instead you might want to know about a symptom, like does a drug reduce seizures or does it improve sleep? You could do that without actually understanding that molecular mechanism. Then we've got these big data sets that exist for researchers to use. So you've got data about the proteins in different people and biological pathways, and then big databases of drugs. And most researchers that work on drug repurposing and rare diseases are going to combine lots of these different techniques all together to find a drug at the end that would go on to be a potential therapy. So like with anything, there's always going to be challenges that come specifically with drug repurposing, as well as its own unique advantages. So a big one is less risk of failure at that clinical trial stage. And that's going to be because they might have already gone through safety profiling. Manufacturing and supply chains are already available, especially if it's a drug that's currently being prescribed a lot. You're already going to have those supply chains in place. Clinical trials will always have better engagement with drug repurposing. That's been shown. It's because of those safety profiles. And then that big one is faster development time. In terms of a few of those unique challenges that you might get, so you might get some gaps in preclinical data. And that would be where you maybe identify a drug that you think would be a possible option to take through to a clinical trial. But when you apply for that, the regulators say, actually, I want you to go back and put it in some more different mouse models or try some more cell assays to fill in some of that gap before you start a trial. Dose and formulation. So you might be looking at drugs that are currently only used in adults. And if you want to use that for a treatment in children, you're going to need to change that dosage. Then formulation might be, you've got a drug that we take orally at the moment, but actually for a certain condition, it might be better off through the bloodstream. And all of those options are going to need to be tested and checked properly. Then a few more bureaucratic things that can come up with repurposing drugs is unlicensed and off-label access and intellectual property can sometimes come into play if those drugs are currently still made by only certain companies. So just to say thank you all so much for listening. I'll be sticking around for the Q&A if you've got any questions, and I hope you enjoyed the talk.